All right, so today's date is the 9th. So if you will click on this, on your side, remember there's up here where mine says published. Yours will say submit assignment. Go ahead and click on submit assignment. And a text box will appear below, and we're going to type the answers in as we talk about this together. So make sure you do number one, and these are all extra credit, so you guys get free extra credit points because we do it together.
Oh, really?
Those that are remote do it on their own. If they watch the video, they'll get the correct answers. If they don't, it's based on what they know. All right.
Basically, it's based on what Google knows.
Well, if they're wise enough to even do that. All right. Number one, what macromolecule is DNA? This comes back from one of our first units of studies. We talked about these big, large macromolecules. There were four of them. We talked about carbohydrates, fats, or lipids, we call them, proteins, and nucleic acids.
Is that right?
Huh?
Is that right?
What?
That's what it said.
What did you say? The acid one. Okay,
Yeah,
nucleic
I
acid.
said the first one. I burned the first one. I don't even know
All
what
right,
I said.
if you know what DNA stands for, deoxyribonucleic acid. So it actually tells you what it is if you know what the DNA stands for. So that's what you'll type in for number one. So you'll put a number one. We talked about monomers of DNA. So what small molecule makes up DNA? So this is a big review. It also starts with an N. It depends on if you have refreshed and relogged into Canvas. If you have to refresh, it's always under attendance.
Let's go, boss.
We're 11.9 at the bottom. All right, so the monomer of DNA is called a nucleotide. And just to help with this next part, there is a 5-carbon, what do we call these? The 5-carbon or 6-carbons? What do we call those? I don't draw a pentagon very well. A 5-carbon, guys, a 5-carbon sugar. So you're going to have a sugar. It's going to be shaped as a pentagon. So that's one of the three parts. It's going to, I forget which where, but somewhere it's going to be attached to. Anybody remember what the little circle represented?
R.
Nope. Phosphate.
She's right.
So phosphate group. You're thinking amino acid.
Oh, okay.
And then this is, there's 4 choices usually, and we call them, technically they should be nitrogen bases. A lot of times we just shorten it and say bases. So the nitrogen base, there are 4 options that we're going to be learning about. All right, so we've answered 1 through 3. So you get at least, once you submit that the last time, because then at the bottom there's a submit button, a submit assignment button again underneath the text box. When I go in to check that, notice it's out of 0 points. So if you get all the points, those are extra credit points. Huh?
That looks so wrong.
nitrogen bases. All right, so we're starting a new unit. Friday at home, you were supposed to have taken your cell cycle test. If you haven't, it has to be taken by anything that we do up through, I forget if it's maybe this week. You have to have completed by November 11th. It's going to be our cutoff date. And you won't be able to make up stuff in the back past that. All right, so we're starting protein synthesis. So I went one step too far. If you will click or scroll down from your attendance, go to modules. If it kicks you out of modules, scroll down. And I just click the arrow to hide the cell cycle stuff so it doesn't get confusing. Protein synthesis. We're going to be talking about protein synthesis first, so I'm going to come over here and we're going to talk a little bit about DNA. Then you're not going to see this part. You're not going to see this part because I'm talking about this part first. Just to review what we've done so far with our first attendance piece. The macromolecule that carries our genetic material is DNA. It's a nucleic acid. DNA stands for deoxyribonucleic acid. And on here, it is divided into 46 pieces. And on these 46 pieces, it carries genes that has the instructions for making our proteins. And they're going to be located, these genes are located at certain points on certain chromosomes. So these pieces of DNA are called chromosomes. We actually have two types of nucleic acids, DNA and RNA. We're going to review DNA, and then we're going to spend a bulk of our time looking at the function of RNA. So you need to know this diagram, a diagram similar to this, what I tried to draw up here. You need to know sugar. For DNA, it is deoxyribose. For RNA, it's ribose. That's where you get their name. The circle is going to just be a phosphate. It contains a phosphate, the element phosphate. We call it a phosphate group. The rectangular piece here is going to be one of four. Please stop talking. One of four, but notice there are five here. In RNA, you don't have T's, you have U's. You have five possible choices. You need to recognize the names, adding guanine, cytosine, thymine, and uracil, but we'll probably refer to them as A, G, C, T, and U. You've got to know this material here. And we'll be coming back to this as well. DNA structure, it's a helix, a double helix. That just means twisted ladder. You've got your phosphate group and your sugar that makes up what we call the backbone, makes up the sides. And then what makes up the rungs or the step of the ladders are your nitrogen bases, and they pair up in certain order. And the bonds between the nitrogen bases are hydrogen bonds, and they're weaker bonds than the covalent bonds that are between the phosphate and the sugar. So with DNA, if you notice, A's, adenines will always pair with thymines. So A and T go together, C and G go together. These are called complementary base pairs, and you're going to be practicing some of that. RNA is a little bit different. It is just a half a ladder, so it's just a single strand. And instead of T's, it has U's, and so A pairs with U, and C and G still pair together. So you're going to need to know how to compare DNA and RNA. The bases are the same with except the T for DNA and the U for RNA. The sugars are different. DNA is deoxyribose, and ribose is for RNA. And the shape, double, it's a full ladder, twisted, where RNA is a single strand. This is going to lead us into our study of genetics. So we need to understand chromosomes. We've mentioned them in cell cycle. Chromosomes are tightly coiled strands of DNA. We only see them as chromosomes in the cell cycle. We have 23 pair in humans, or a total of 46 individual. You get 23 from mom, you get 23 from dad. They give you an example up here for dogs. Dogs have 37 pairs, so you have 74 total. You get 37 from mom, 37 from dad. So different organisms have a different total number.
Does that mean they're technically smarter than us?
No, this means they have more
Yeah, see that
genes.
one.
All right, so this is kind of a chromosome. This is the centromere. So when it has been copied in the cell cycle, they're joined together. So one piece of this is a single chromosome. And on this chromosome, it is divided into segments, and each segment controls a gene. So a gene is just a piece of DNA. It has instructions for typically protein. And so you find DNA in the nucleus. So here is a chromosome that's unwound. It's a really long piece. And certain pieces of that will code for certain proteins. So we're going to review here replication, and this is sort of where we're going to practice today. DNA has to replicate. Does anybody remember what part of the cell cycle it replicates? I'd look up on the board.
Which one?
The electronic board.
Oh.
S phase,
Yeah.
that is part of interphase. So before the cell can even think about dividing, it has to have a copy of DNA. So DNA replication, we didn't talk a lot about that in cell cycle. We're going to talk about it now and how it makes copies. So here's kind of the steps. You're going to be watching a video on this and then doing an activity. So it's going to unzip first. It has special enzymes that will come in and break apart the bonds between the bases right here. Then it has other enzymes that will help bring in the bases to make the new strands. One strand, the leading strand, is made in one direction, and the lagging strand is made in a different direction, and it's kind of made in chunks. So it's a little more complicated on the lagging strand. Semi-conservative, because you get an old strand and a new strand with the two new pieces. That's why it's semi-conservative. And we're going to practice some of this base pairing. So when we go back to here, you're going to have a video on DNA and replication. For your practice, we're going to look at this. The video is just your ed puzzles you're familiar with. When's the last time you've logged into your Chromebook? Yes, you do. You don't have all the other stuff because I haven't made it live. All right, so here are some questions based on what we've talked about and what the video will talk about. And you're going to answer these questions. And then I wanted to look down here at this part right here. To replicate. So if you want to scroll down to number 2, we're going to do A together. As soon as I get it written up here. C-A. C-G. No, 2 C's. No, wrong one. C-G-T. CC, AGA. Got 4 T's at the end.
Yeah.
All right, so DNA replication means you have to have 2 strands. This is 1 strand. So what you're going to do here is you're going to pair up with what it's supposed to pair up with. So what did we say A would pair up with? A pairs up with T, C pairs up with G, and it can be read the other way. So if it helps you, like this. So we're going to do 12A together. So you'll need a text box. So if I see an A, what am I going to type?
T.
T. And it doesn't matter if yours doesn't line up exactly underneath the letters, as long as they're in the same order. I see a T, what am I going to write? A. I have an A, I'm going to write
W.
T. C, I'm going to write
G.
G.
C.
C.
G. G. B.
Hold on, too fast. A is a T. G is a T, and then A's. All right, so that is what you're going to do for 12. Before you try to answer the rest of these, you may want to watch the video. So if you just go back to previous, watch the video first, and then attempt to answer the questions so that I can help you answer the questions. Just so you know, Thursday, I will be observed in here, so you'll want to make sure you're following along and doing what I'm asking you to do. And part of the activity that we'll do Thursday will actually be on paper. So wherever you've left your little pencil pouch in whatever class, make sure you bring it Thursday. Or I'll try to have a pencil for you. But if part of what we'll do will be on pencil on Thursday. So you may want to start with the video unless you want to finish up number 12 first, and then go back and listen to the video before you answer the other questions. I'm going to stop the recording.